Namaskar. Today we will study the M1 receptor. M1, M stands for muscarinic receptor, and muscarinic receptor are of 5 types: M1, M2, M3, M4, M5, and we will discuss today M1 receptor. And M1 receptor is located in the gastric paracrine cells, the parietal cells of GIT, in CNS, mainly in the cortex and hippocampus, and in the neural, that is in the ganglia, autonomic ganglia, and in the enteric part. So, this M1 receptor is a G protein coupled receptor, and it is a type of GQ type of G protein coupled receptor, where the second messengers are IP3 inositol triphosphate and diacylglycerol, and the agonists of M1 receptor, example oxotrimorin is the agonist of muscarinic receptor. Antagonist is M1 selective antagonist is pyrangiapine and telangiapine. And before you know how this receptor functions, let us see GQ type of G protein coupled receptor. And you can see in the GQ type of G protein coupled receptor, the effector is phospholipase C beta. Phospholipase C beta is activated by the alpha Q. As we know, G protein coupled receptors have the subunits alpha, beta, gamma, and alpha Q, when binds with the phospholipase C beta, it activates this enzyme phospholipase C, which converts phosphatidyl inositol diphosphate into IP3 inositol triphosphate and diacylglycerol. IP3 acts on the sarcoplasmic reticulum to release calcium, and calcium binds with calmodulin to form calcium calmodulin complex, which activates protein kinases to produce the cellular effects, mainly contractions of smooth muscles, and in this case in acid secretion is also caused by this calcium, that we will discuss how. And then diacylglycerol activates the protein kinase C, and that activated protein kinase C causes protein phosphorylation, and hence there is cellular functions. So now we will see how this M1 receptor functions, and we will give the example of the M1 receptor present in the parietal cells of GIT, and its function is acid secretion, and how acid secretion is caused by acetylcholine by acting on GQ receptor. As we know, the acid is secreted into the lumen. So if it is the parietal cell, this is lumen. Into the lumen, the acid is secreted through proton pump. We call it as the proton pump, or H plus K plus ATPase. This enzyme is also called as proton pump, and this is a symport called potassium chloride cotransport or symport, and here from the cell, acid or proton is pumped in exchange of potassium, and this is a co-transport process where potassium and chloride both come from the parietal cells into the lumen. So there is H plus Cl minus, that is the gastric acid. Acid is secreted, and how this is there due to the depolarization of the parietal cells, and this depolarization is caused by calcium. So when acetylcholine binds to the M1 receptor, which is a GQ type G protein coupled receptor, the effector is phospholipase C beta, which is activated, and this phospholipase C converts phosphatidyl inositol diphosphate to IP3 and DAG. IP3 helps in release of calcium, and this calcium causes depolarization, which causes acid secretion. And in the CNS, it causes CNS excitation, and that is again through the GQ type G protein coupled receptor, that is M1 receptor. So this is all about the mechanism of M1 receptor, which is a GQ type G protein coupled receptor. Thank you.